Today's video is about xylem and phloem, the transport vessels of plants. The two dot points covered by this video describe current theories about the processes responsible for movement of materials through plants. And there's a first-hand investigation where you are asked to draw transverse and longitudinal sections of both phloem and xylem tissue. Xylem and phloem are called conducting vessels. Xylem is transporting water and minerals, and phloem transports sugar. In young plants, xylem and phloem are found within vascular bundles, which you can see here, and they're separated by something called cambion cells. Cambion cells divide and differentiate into xylem and phloem. Just having a look here at the xylem cells, you can see that they are semi-hollow tubes, remembering they are dead cells, and they have these perforations at the ends of their cells that allows the water to flow through. In phloem, you have a similar sort of thing where you've got sieve plates in between the cells, but you also have these special companion cells here that are found next to the sieve plates. So pay attention to those as we go through and look closer at xylem and phloem tissue. So in this slide you can see the cross sections of the root, the stem and the leaf. And you can see that the organisation of xylem and phloem changes as you travel throughout the plant. So in the root there is xylem in the centre forming a kind of X, so that's a good way to remember. And then the phloem is outside that. In the stem here we've got those vascular bundles we were looking at before, and the xylem there is in the towards the centre of the stem, and the phloem is towards the outside of the stem. You follow the purple colour here for xylem. And then when it gets to the leaf, you have the xylem's actually on the upper side of the leaf and the phloem is underneath. Now if you learn that, you can find it easier to identify the different types of vessels in a diagram or in a slide that you're looking at. And again, you can see here different representations of where xylem and phloem are located in the root, stem and leaf. So here are the two current theories. For xylem, the theory is referred to as transpiration stream or cohesion-adhesion-tension theory. There are animations that you can watch. I recommend that you look some up and watch them. For phloem, it's called the pressure flow theory or the source path sink theory. It's also referred to as sugar loading or phloem loading. And translocation is the common name there, whereas it's transpiration for xylem. The transpiration stream theory of xylem and water moving through it goes like this. So it's called a transpirational pull. So it's started by water evaporating via the stomata in the leaves. So at the top of the plant, transpiration is happening. And as that water evaporates, it basically sucks water up through the xylem vessels like a straw. And this is because water has particular properties of cohesion. So water molecules like to stick together. That's what cohesion refers to. And adhesion is about how water likes to stick to walls of vessels. So between cohesion and adhesion, you've got water getting pulled up via its stickiness to itself and to the sides of the xylem vessels itself. So water enters the root by osmosis, and transpiration or pull draws water up the stem. So this is a passive transport. No energy is required by the plant for water and the dissolved nutrients to travel up. It only flows upwards, and we know that about xylem already. In summary, it is the cohesion and adhesion of water molecules that makes transpiration a successful passive process. You can see the xylem tubes here are hollow dead cells joined end to end. They form a tube like a microscopic straw. The movement of materials in phloem, understanding that phloem's about sugar and getting sugar from the leaves where it is produced via photosynthesis to all of the other parts of the plant that need to use the sugar to perform cell processes via cell respiration. So let's have a closer look at this diagram here. So we start at the leaf, and sugar is actively transported into phloem from the leaves, and as that happens you get a concentration gradient forming, so there's more solute in the phloem, so water follows that sugar by osmosis. So you have active transport of the sugar into the phloem and passive transport of water. As that water flows in, it pushes the sap. It creates a positive pressure potential that causes the sap to flow within the phloem. We call the leaves the source because that's where your sugar is in high concentration and is created. And when the sugar gets to cells that need it, where growth is happening, then we get to what's called a sink, somewhere that the sugar is needed. And the sugar is then actively transported out of the phloem sieve tube cells into those areas that are needed. The roots are one of the sinks here, and water follows those by osmosis as well. So you're going from an area of high sugar concentration to an area of low sugar concentration, but this is an active process. It's active because it's against a concentration gradient. Sugars are transported to where they're already concentrated in those sinks because energy is needed, so energy has to be used to achieve that. This is active transport, and so you get the companion cells that are next to the phloem sieve tubes have many mitochondria to produce the ATP, which is used to power that active transport. So you need to complete an investigation where you examine and draw sections of phloem and xylem tissue. So before we go into that, we might have a look at the types of cells that you need to be able to draw. So xylem has 3 types of cells. We've got the vessel members, also called vessel elements, tracheids, and fibers. The fibers don't actually transport water, but the tracheids and the vessel elements do. So if we look at the vessel elements, they have distinctive pitting on the outside of them, and in between each of the cells there's the perforation plates, whereas tracheids have these slanted ends on them. For phloem, we have sieve elements, or sieve tubes they're often called, and their defining feature is this sieve plate, and next to them we find companion cells. You need to draw both transverse and longitudinal sections, and this is a slide showing you what the difference is between how you cut to achieve those slides that you can draw. Transverse meaning across and longitudinal meaning along. Here are some pictures of slides, just so that you can identify what you're looking at when you create your own slides. Your own slides are not likely to be as clear as this, but these can give you a good sense of what you're looking at. So when you slice through a root, you're going to see these sorts of cells, these big round cells with thick walls. They're going to be your xylem, and your phloem are much smaller next to them, and they do stain differently. When you cut through a celery stem, and this would be the outside of the stem here, then you can see these vascular bundles, and again the large holes with a thick wall around them are going to be your xylem tissue, and next to those is going to be your phloem. And here is a leaf with the xylem at the top, and again those big round heavy walled cells, and the phloem smaller beneath them. Here are some longitudinal sections. So the phloem and the xylem are right next to each other. You can see the xylem here and phloem here. And these next few slides show you some ways of drawing them, because that can be a bit difficult to draw from your slides. So here are some examples that you can use to help you. These are tracheids. These are vessel elements. And here I'm detailing what you need to include in a xylem drawing. So I've got a transverse section drawing here and a longitudinal section drawing here, which when you draw you need to indicate that they are transverse with these sorts of labels here, TS and LS. In the transverse section you're cutting through the cells, so you're going to have some small xylem vessels that are called tracheids, and you're going to have those big, large, hollow-looking vessel elements. But when you draw in longitudinal, you need to indicate the perforated end plates and the lignin rings. This is an electron micrograph of xylem vessels. You can see the rings of lignin quite clearly here, and these are vessel elements. In phloem tissue has two types of cells, the sieve tube cells and the companion cells. Within the sieve tube cells you have these special end points called sieve plates, and within the companion cells you're going to have a visible nucleus and quite possibly mitochondria. So including in your phloem drawings. You can see in the, okay, they're the wrong way around. This is the transverse section here. And you can see you've got your sieve tube cells and your companion cells. The companion cells are right next to the sieve tube cells. Remember, they provide the energy for the sugar loading. And you're going to note the nucleuses, a vacuole, maybe cell wall and cell membrane. And then in longitudinal section, you're going to draw at least 2 sieve tube cells, showing that perforated plate. The sieve plate, and next to them you're going to have the companion cells. Now these little black marks there are called plasmodesmata, and as you can see, the companion cells are smaller than your sieve tube cells.